Okay, in this video, we're going to be talking about action potentials. How is a signal actually transmitted along a neuron? Remember, a neuron is an individual nerve cell. So how is a signal sent? And if you remember, I talked about it's along the cell membrane, that the nerve cells have specialized cell membranes. And so this part of our body is, well, it's kind of electrical. Sometimes people think that we run on electricity. Often their misconception is when you use a defibrillator to jumpstart somebody's heart, that you're jumpstarting their heart. You're literally like jumpstarting a car. You're sending electricity to their body that makes their muscles beat. That's not how it works. We're powered by heat. We actually, the thing which electricity, there's no batteries in us. There's nothing like that. We're powered by heat. So for example, ATP is not electrical energy. Sometimes, you know, ATP is shown like those phosphate bonds at the end there are shown with like sparks shooting out of them. No, it's heat. When those bonds break, it basically it increases the vibration of the molecules around it, and that permits chemical reactions to happen. What is electricity? Electricity is when you have a potential, a charge potential. A potential is kind of like a concentration gradient where you have, you know, a positive charge over here and a negative charge over here. That's a potential, and electrons will move across that, and that movement of electrons from across a potential can be used to do work. Sort of how neurons work. But it's not electrons that actually are moving. It is ions, salt ions, that are moving. And they're positively charged, and there's some negatively charged salt ions as well, but there's a difference in potential across the cell membrane. So one area of the cell membrane is positively charged relative to the inside of the cell membrane, essentially. And the flipping of that charge is how a signal is transmitted. Okay, so let's talk about, so there's two types of impulse of how information essentially is transmitted across nerves. So number one is saltatory transmission, and that's basically, saltatory means salt transmission. And so that's these ions, the salt ions. That's why you need to eat a certain amount of salt. You know, Mr. K likes his fries, and he likes salt on his fries, and everybody's like, oh, your blood pressure is so high, you shouldn't eat. Well, you know what? I need my nerves to work, so I need that sodium chloride in the salt. So the second kind of transmission is synaptic transmission. And so basically synaptic transmission happens in the space, it happens at the end of an axon and the start of a dendrite. So there's an axon, there's a dendrite, and so signals are going to be transmitted across, and it doesn't work on salt. It works on something called neurotransmitter, which are basically like molecules that do like a lock and key kind of thing. They're related to hormones, and we'll talk about that next. So first, let's talk about this action potential. There's a lot to unpack in saltatory transmission. So normally, you've got a nerve, and it's hanging out, and there's nothing happening, okay? So what the condition is, that is called the resting potential. And basically the outside, the extracellular space around the nerve is, and pretend this is the axon. We study axon, so this is the axon. Remember, axon is impulses moving away from the cell body. But it could be also a dendrite, where this is moving towards the cell body. But so, because the axon is the thing that was studied, we're going to see words like axoplasm, okay? Axoplasm is just cytoplasm of the axon. And if you need to review what an axon is, here it is. There's the cell body, there's a motor neuron, there's a big long axon. Here's an axon in a sensory neuron. Oh, you guys can't see that. In a sensory neuron, here's the axon, a sensory neuron, but here's the dendrite, and these are the Schwann cells. There's the nodes of Ranvier. So this action potential is going to be happening at these nodes of Ranvier. The man they call Ranvier. So that's where we are. So in a resting potential, you have positive charge out here and a negative charge in here. The difference, okay, is 65 millivolts. Okay, it's measured as 65 millivolts. Now, how does this resting potential exist? What we have essentially is, I'm just looking at this picture here. Do I like this picture? Oh, because it's this. Okay, I see. Okay, so this is the extracellular fluid. This is the axoplasm or the cytoplasm. And what we have in here is, so we've got a lot of sodium ions. Sodium is positively charged, okay? So we got a lot of sodium. Now, we've got some sodium ions in here too. And we also have potassium ions, and the potassium ions, the majority of the potassium ions are in here. And now you're saying, well, wait, these potassium ions are positively charged. And I'm going to tell you that, yes, they are. You are correct. And the sodium ions are positively charged. So how is there a potential across here, positive to negative? Well, in addition, and we don't have it on any of these, we don't have it on these the diagrams here, but in addition to potassium ions being in here, salt is made of sodium chloride. There are chlorine ions here, and chlorine ions are negative. And the chlorine ions don't do anything. They don't move across the membrane. They don't do nothing. They just are in here being their negative selves. And the negative chlorine ions cancel out the potassium ions that are in here. And there are more also sodium ions. The concentration here is greater than of potassium ions. So basically you got a lot of positive ions out here making a potential difference of 65 millivolts. Okay, that's a resting potential. You also have here, the membrane is specialized. You've got channels. You've got all integral proteins. Remember from our membranes unit, everything comes back. Everything comes back. I told you all of this stuff comes back. There's ionic bonds. These are dissolved in water. All of that stuff should be in your mind right now. But so we have these peripheral proteins here. And some of these peripheral proteins are channels for sodium. Some of these are channels for potassium. And there are also sodium-potassium pumps. So remember, channels are passive. Those are going to be going with the concentration gradient, not using ATP, passive. But then there are sodium-potassium pumps along here as well, and they use ATP. They move against the concentration gradient. And what they're doing is they're taking potassium, putting it in here, taking sodium, and pumping it out to the extracellular fluid. Potassium, sodium, potassium. I think you get 2 sodiums for 1 potassium, but don't quote me on that. So, here we go. What happens during... Now, what's going to happen during an action potential? Well, so let's say you have the thought of, I would like to do this. I would like to contract my index finger. There are muscles actually in here. They're not up here, sorry. There's muscles in here attached to a big long tendon that runs up here. You can watch, you're old enough to watch The Terminator from 1984, and he's like a cyborg, and he operates on his arm. It's like an Arnold Schwarzenegger movie. You guys don't know who that is, but it's okay. And he like cuts open his arm, and then he moves the machinery in here. It goes. Anyways, it's a really cool movie. So we have the same thing, except it's the tendon. There's no metal in here, and you have muscles that contract here that can move, that attach the tendon and move the tendon. You can even feel the tendon moving in here. I can even, if I press on the tendon, I can make my finger move a little. So anyway, so you decide you're going to move that, you're going to do that to the finger. You're going to do this. And so you send a signal from the motor part of your cerebrum, and it goes down, and it goes down your central nervous system, and goes down, this impulse goes down the spinal cord, and it exits at the right place. And then the stimulus then stimulates the dendrite, which is like, okay, the way nerves, these nerve fibers work is they're what's called, they have all or none firing. So there's a threshold of stimulus that has to happen in order for the cascades to happen that will actually send a nerve impulse here. And once the nerve impulse is sent down, it starts, it is exactly the same every time. There's no degree of nerve impulse. So there's no stronger or weaker nerve impulse. What is meant by a stronger or weaker signal is that you engage more individual nerve cells. So, for example, if you go to the gym and you are lifting weights, the amount you send a signal, let's say this, we're going to lift this speaker up, and we're going to lift this speaker up. And so I'm sending a signal to lift this speaker. I'm putting pressure on this speaker right now, but I'm not lifting it up because I'm not sending enough signal to engage enough motor neurons. So there's not enough transmission for a larger group of motor neurons to lift this speaker. But I can increase the signal by increasing the amount of motor neurons that fire, and now more individual muscle fibers are contracting, and the muscle fibers are the things that do work. So remember, a motor neuron, it ends up and it signals a group of muscle fibers. So now I'm engaging more muscle fibers to contract. So more muscle fibers are contracting in my muscle here. I guess this is going to be a bicep here, and I can lift this thing up. But each individual cell, this just fires all or not at all. And the more cells you fire, the greater the signal. So that also works the other way for sensory cells. The more cells that are fired, the greater the stimulus, the greater the signal. Remember, we talked about that reflex arc. When you touch a hot stove, many now sensory cells are engaged and are firing, and they're all firing exactly the same level, but they're all firing, and it's enough to engage those interneurons, which will then signal some motor neurons to contract your arm. Okay. So how does this happen? So you have to reach a threshold stimulus here at this end, basically on the dendrite. You have to reach a threshold. There has to be enough stimulus for this now to fire the neuron, the individual neuron. And so what happens is, initially there's a stimulus, it reaches, there's even a threshold that is reached. So here it is, there's the stimulus, the threshold is reached, and then what happens is something called depolarization. Sodium channels in the cell membrane, in that axon cell membrane, or let's go look at it here. So here's a close-up of it. So these sodium channels open. That's the first thing to open. And there's a greater concentration of sodium out here than there is in here. So there is a concentration gradient. And what happens, guys, when you have a concentration gradient? You get diffusion. And this is assisted diffusion down these channels, because these are channels. And so sodium rushes in to the axoplasm, the inside of the axon. And now there's potassium here and sodium and a whole bunch of sodium together. So those chlorine, those negative chlorines are getting canceled out. Now, I mean, they're getting overwhelmed by the positive charge. So what happens is the potential flips because there is more sodium and positive sodium and positive potassium in here. There's very little sodium now in here. Out here, it's starting to equalize in here. This has an overall, even though it's not actually negative, but compared to here, it is negative. And once it reaches out here, negative, I believe it's negative 40 millivolts. Oh, it says here negative 30. Is it? I've always read it was negative 40. Potassium gates, all are non-response. Well, let's not worry about it. We're just, it says here 30. Okay, let's just say 30. Once it starts to reach around 30, 40 millivolts, what happens is that triggers potassium channels in the membrane to open. And now there's sodium's all in here, but now the potassium's there's a lot of potassium in here. And what happens is the potassium channels open, and so the potassium starts to diffuse out into the extracellular fluid. And what happens now, so now positive charges are heading out. The sodium is still in here. I mean, when did these sodium gates close? So the sodium gates have closed. Basically, we've switched. We've closed the sodium gates and we've opened the potassium gates. So sodium gates are closed, potassium gates, and here's outside, inside. So now potassium on this one, of course it's upside down to the one before. But anyways, so now potassium. So basically the potassium and the sodium are flipping, and as they flip, the resting potential is restored, except it's different. So we're repolarizing is what we call, and it's different. And actually, look, it goes way over. It goes to like minus 90. All of these, look at this, the resting potential, because we were saying it's negative 65, and here it's minus 70. So this maybe nobody knows. I was saying it's minus. I've read minus 65 3 or 4 times now. But anyways, it goes beyond. So all the potassium runs out, and now the sodium ions are on the inside, the potassium ions are on the outside, but we have the same resting potential because those chlorine ions never went anywhere. They're still on the inside, so now instead of canceling the potassium out, they're canceling out the sodium, so we've got negative, we've got, I guess, more stuff went out. And right now, even though we've restored resting potential, this area of the axon cannot fire again. So what this does is it because firing, again, is the sodium moving in, right? Depolarization is sodium. Sodium moves in, if it goes inside, sodium channels close, potassium goes outside, the charge, the potential, the difference gets restored. But the signal can't go backwards is basically what happens. Because look, so you've got, it starts here, the sodium, the sodium goes in from the outside, and that action actually, it spills over and it starts the next region from the sodium moving in. But now and then, when the repolarization, all the potassium is out there, it can't go backwards because potassium, the channels go, it needs to be sodium out here for it to start, is essentially what it is. So look, there you go. There we're right. Then we're repolarizing, so the potassium is going out, and that starts the sodium, that starts the sodium going in here, and then this next region. And so that's how the impulse travels down. It's just sort of all these channel membranes opening and closing. So first you have the sodium membranes opening. It reaches a certain voltage of potential. You know, over there it says minus 30 or plus 30, and I'm saying it's 40. So let's call it even 35, and that triggers the next area to start the channels opening. Meanwhile, the potassium channels are opening, and the sodium channels are closing. And you get it. Now, what happens next? So now these guys are flipped. Potassium's on the outside, sodium's on the inside. We have the right potential, but we've got the wrong ions. So what happens is then the pumps start up. Have you ever been someplace where there's pumps, like you know, like a boat or something that gets filled up with water? Or if you've ever been like to a place that uses like groundwater for their toilets and stuff, and you flush the toilet and you hear the pump go and start pumping. Well, that's what's happening here. So we've got the right resting potential, but now, and we're actually, this is called hyperpolarization. But now what we're going to do is we're going to restore sodium and potassium concentration. We're going to put the sodium on the outside, and we're going to move the potassium on the inside. And so we use that with sodium potassium channels, active sodium and potassium pump. So then there they go, and they're restoring. It's still the resting potential, but now the correct ions are where they're supposed to be. And you can see here, it actually shows it. So it's like more sodium moves than potassium. The potassium is, the sodium is a greater charge. Yeah, there we go, look, 40. Potassium gates open at the same time as the sodium gates close when the cytoplasm becomes plus 40 millivolts relative to the outside of the cell. So when it's plus 40 millivolts, that's the cytoplasm has both sodium and potassium on the inside the cytoplasm. And when those potassium gates open, that concentration of potassium is greater on the inside, so that potassium goes out and restores the resting potential, and then the sodium potassium pumps flip them. This does not require ATP. These gates, the gates don't require ATP. This is passive transport or passive diffusion or assisted diffusion. This does, because it's a pump that uses ATP. And so you see, you can't, the action potential, this signal moving down, it can't be greater or lesser. It's just ions moving across the membrane. There's no more ions or less ions that move. It's like all the ions and none. So it's all or none. The response is all or none. How you control the strength of the response is how many of these neurons fire in a bundle, basically. How many in a bundle will fire? More neurons, more motor neurons firing, the stronger the pull of the muscle, because more muscle fibers are engaged. Your muscles are a combination of muscle fibers that contract. The more muscle fibers are actively contracting, the more power, the more motors basically are running to move that muscle. The brighter the light, the more sensory neurons that are connected to your eye are now firing and reading that there's a bright light going. So that's how the brightness of the light, but all of them are just doing this, it's just these ions moving along. Okay, so this was supposed to be a short video, I'm sorry. Next, synaptic transmission. So we've got a couple of places. So motor neurons, they start in your spinal column, and they travel all the way right out to your muscles. But between the spinal column, and the dendrite of the motor neuron, and vice versa, the sensory nerves that runs all the way in, and the spinal cord, there is a space. So you've got this action potential, this salt, you know, the sodium going in, potassium going out, and then them restoring again. And once it reaches the end, what happens at the end? The membranes aren't continuous with the next nerve cell. So what happens at the end is, instead of there being sodium out here, what happens is as this action potential reaches here, it activates channels that are for calcium. So there'll be calcium out here instead of sodium, calcium ions. And the calcium ions go in, because of the concentration gradient, they pour in. And there are a number of vesicles called synaptic vesicles. We learned about vesicles, you know, these would be made by the Golgi body. And these are the type of vesicles that hang out near the cell membrane until they are called for and released. They're that kind of vesicle. Remember those vesicles? So you know, these neurotransmitters are made in the proteins made in the ER, or lipids made in the smooth ER. Lipid hormones made in the smooth ER. They get packaged in the Golgi body, and then they get transported to here, and they hang out here. And they're attached, here it's got, this protein is kind of wrong. These are called docking proteins, or there's another name for these proteins, which I liked better, and now it has completely escaped my, getting old. Contractile proteins. There we go. Contractile proteins. So basically there's these contractile proteins, and imagine them, they're like a finger or like a rope. And the vesicles are attached at the end of the rope, and then the other end is attached to the cell membrane. And what happens is calcium pours in here, and calcium changes the shape of that contractile protein. And what it does is it contracts, that rope contracts, and the vesicle slams into the membrane here, the cell membrane, and fuses with the cell membrane, and you get exocytosis of the neurotransmitter. A whole ton of neurotransmitter gets poured out into what is called the synaptic cleft. And this neurotransmitter diffuses across the synaptic cleft, and on the other side of the synaptic cleft are receptors, special peripheral proteins that are lock and key, and the neurotransmitter fits into those receptors, and then those receptors start an action potential. So that's saltatory, you know, sodium pouring in, and off it goes down the next nerve. This slows things down, obviously, because it's relying on diffusion, vesicles merging, so it slows things down a little bit. It also is a place where things can mess here. So what happens is these bond, and then enzymes come along and they eat all the neurotransmitter. They break down the neurotransmitter because you don't want this signal being continuously sent. They eat this neurotransmitter. You have a lock and key thing here. You've got enzymes that eat neurotransmitter. Drugs. Basically drugs, a lot of drugs work here. Drugs come in here, they pretend to be the neurotransmitter. Maybe this goes to, you know, makes you feel good, makes you feel happy. It's an action potential that makes you feel good. So drugs, they stimulate this and you now feel better. But most likely these drugs, they don't fit exactly right into the receptor, and they break the receptor or wear the receptors out. So then your neurotransmitter, when you actually get your neurotransmitter, it doesn't work as well. You don't get the good feeling naturally. You need the drugs to make the good feeling happen. Drugs that block that enzyme, inhibit that enzyme that eats the neurotransmitter. So you basically get a longer feeling of whatever this feeling is. This could actually be also depressed, to depress a signal to depress certain organs and certain things. This is a lot of this is working in your autonomic nervous system. We'll talk about next. So a small inkling into, you know, some of the stuff that you can short circuit this. Now, there's a question somewhere, why can't the signal go backwards? Quite simply because this is called the presynaptic, so this is called the synaptic cleft, or synapse. This is called the presynaptic membrane, and this is called the postsynaptic membrane. The presynaptic membrane has those contractile proteins, the vesicles. What it doesn't have is these receptors. So the signal can't go backwards because the neurotransmitter's got no receptors to get recepted in the presynaptic thing. So they can only go here. This also can't produce vesicles because it doesn't have those things. This also can be like an organ. So at the end, you know, and muscle cells and stuff like that. So you've got an axon here, ending here, and this could also be glands to secrete stuff. Muscle cell to contract. You got these receptors. That's basically what these neurotransmitters do. They're like the final stage of your intention. And they can be hijacked by things that pretend to be the neurotransmitter, or things that destroy the neurotransmitter, or things that destroy the enzyme that destroys the neurotransmitter. All kinds of things like that. That's it. Presynaptic, postsynaptic. And you can have, depending on what axon this is, from what kind of cell this is, you can have neurotransmitter that, if this is a gland, you can have neurotransmitter that inhibits this gland. Like this receptor is like, stop doing what you're doing, gland. Stop making the thing that you're making. Or you can have neurotransmitter that is excitatory, so inhibitory and excitatory, and it tells the gland or the organ to do more of the thing that it's doing. Okay. Anything else? No. So review. Two kinds, action potential along the membrane. You got three kinds of integral proteins here. You have sodium channels, potassium channels. Sodium potassium pump does both, moves both. It actually swaps. It takes a potassium from the outside, moves it to the inside, and puts sodium out. Resting potential, lots of sodium on the outside, potassium on the inside, along with chlorine ions, which are negative. So there's a difference of potential, different 65 millivolts, we're saying, because just there's more positive ions versus positive ions and negative ions here. Threshold is reached. Sodium pumps open, those integral proteins open. Sodium pours in, and now you have sodium and potassium inside, and no sodium out here, so you got more positive charge on the inside. Once it reaches, the inside reaches positive 40 millivolts, or 35, let's say 35, the sodium channels shut, the potassium channels open. Potassium now, along concentration gradient, this is all so far a concentration gradient, starts to go out. Resting potential is restored. That 65 millivolts is restored. And the potassium ion pumps start. Because it's potassium out here, sodium in here, we can't have the signal go backwards. It can only, that impulse will only go forwards because it's just opening those sodium pumps. The sodium channels. Meanwhile, here, sodium-potassium pumps are putting everybody back to where they belong. Synaptic transmission, so that action potential travels the sodium and the potassium stuff, but then it hits these integral proteins. These integral proteins don't care about sodium or potassium. Instead, they take calcium from outside, pump the calcium in. There are vesicles with neurotransmitter close to the presynaptic membrane, and they are connected by contractile proteins. The calcium attaches to, there's like an active site on those proteins. Calcium joins that active site, changes the shape of the protein, so they slam those vesicles into this membrane. They fuse. There's probably some proteins in the vesicles and in the membrane here that, this exocytosis gets coordinated with the cytoskeleton. Neurotransmitter diffuses across the synapse into receptor proteins, peripheral receptor proteins, and these will do something. These will trigger either a new an action potential to continue on down another nerve, stimulate an organ or inhibit an organ to do something, stimulate endocrine or endocrine glands or exocrine glands to secrete stuff, or inhibit them from secreting stuff.